Welcome back, friends. Welcome to another video tutorial from Shomu's Biology. We've been talking about the nervous system of human body, and we're looking at the physiological aspects of the neuron. We saw the anatomy of the neuron, and we also saw the different ion channels that are present across the neuron. I told you in the last video that the ion channels play a vital role in maintaining the membrane potential, that is a voltage across the cell membrane of a cell. Now all the cells, especially if you look at for the neurons, they have a resting membrane potential, that is a baseline of membrane potential to maintain. Now gradients cause the ions to move across the cell membrane. Like normally ions move from the high concentration to the low concentration. So due to this gradient concentration change, the movement of ions are also varied. That creates this voltage across the membrane. For example, if I draw it here, if this is the cell membrane inside and outside, and if there is a net flow of sodium ion outside the cell, there should be a positive charge outside the membrane and there should be slightly negative charge inside the membrane. So these are the type of modification that take place. The electric potential is called the membrane potential, that is formed due to the distribution of those ions, which is the result of the net movement based on the gradient of the ions across the membrane. For example, normally if you look at these nerve cells, you'll find out that there is a slightly positive charge outside the membrane and slightly negative charge inside the membrane towards cytosol. Due to this net change, there is a electric potential formed across the membrane, and that is normally belonging to minus 70 millivolt. That is known as a resting membrane potential. Now the question is how this resting membrane potential is established. As I told you, it's due to the opening and closing of ion channels, but obviously we need to go into the details of how ion channels open and close to produce this minus 70 millivolt membrane potential. Now the question number 2 is how the cell maintain this minus 70 millivolt of membrane potential. So these are the two things that we need to learn, because we know that in nerve, when there is an alteration of this membrane potential, that allows the information to flow from one neuron to the other neuron. For that, this membrane potential is altered, is changed from minus 70 millivolt. Let's say it can be changed to minus 90 millivolt. That will also allow the membrane potential to change, and that will involve in transferring the nerve impulse. So let's look at the process. So the goals for our learning of this chapter is to know the relative concentration of ions inside and outside the cell, to recognize that cells have selective permeability of ions, and to understand the equilibrium potential for potassium ion, to understand that sodium and potassium determine to control and start the resting membrane potential, and also to understand the role of sodium-potassium ATPase pump that maintains the resting membrane potential. So let's look at each of this process. Start with how the membrane potential originate in the first place, and secondly, how to maintain the membrane potential. To understand membrane potential, the first thing we need to know is about the ion concentration and the selective permeability of a cell membrane. The first thing we'll understand is the change in ion concentration across the cell membrane. This change in ion concentration actually drives the movement, the net movement of ions. If you recall earlier, we talked about the intracellular concentration of sodium, potassium and chloride differs from the concentration of these ions that are present outside. Inside the cell, the concentration, if you look at here in this picture, this is inside, this is outside. So my right hand side here in this picture, the intracellular side of the cell, they normally contain the concentration of the positive ions are high. It is balanced by high concentration of negatively charged proteins or other anions. Negatively charged proteins can be this bigger one, or it can be anions like chloride. It is balanced in this fashion. While in extracellular fluid, if we look at this side, the concentration of positive sodium ion is high. It is balanced by high concentration of negative chloride ions. Okay. So potassium is more inside the cell, sodium is more outside the cell. And this distribution of ions are balanced by anions that are there. So anions are not the key players that alters the membrane potential, but that helps to maintain the ion gradient across the membrane, which is also very important stuff. Now, alongside of this gradient change, we also need to understand another important fact, that is the cell membrane's feature of selective permeability. Ions are not soluble in the lipid bilayer, so they cannot easily diffuse through the membrane. They need channels or helper proteins that will allow them to pass and move. If a cell has channels for a particular ion, we say it permeable to that ion. Since most cells are permeable to some ions but not to others, they exhibit what is called as selective permeability. For example, one ion channel, that is sodium channel, it will allow sodium to pass but not potassium to pass, right? So a cell contains more sodium channels will help to move and migrate sodium across the membrane. The other cell lacking sodium channels will not be able to do so. So that is a selective permeability. Now the cell is permeable to potassium. Many cells in the body selectively permeable only to potassium, because it's a must to have a permeability to potassium and sodium normally to maintain the resting membrane potential. Now excitable cells are very permeable to potassium. If a cell is excitable, that should be very much permeable to potassium and slightly permeable to sodium. And if you look at neurons, they are selectively permeable to potassium. So in case of neurons, if you look, they're selectively permeable to both potassium, sodium and chloride ions. As we saw earlier in the picture, you see, the gradient of all these ions are listed. While sodium, more outside, less inside. Potassium, more inside, less outside. Chloride, more outside, less inside. Cells are impermeable to any kind of negatively charged proteins or other large anions found inside them. These ions are too large to pass, so that is another way of selective permeability. This picture actually depicts the selective permeability very well, because lipid soluble molecules can easily diffuse through the lipid bilayer without any issue. They don't need any extra help or any other carrier or channel proteins to present. But non-lipid soluble molecules are prevented normally to diffuse to the lipid bilayer, but non-lipid molecule can also pass through the membrane if they have a membrane channel. That is the importance of a membrane channel protein, and we've already talked about the membrane channel protein in the last video. So if you don't know about channel proteins, I should strongly recommend you to go and watch that video so that you get much idea about this video. Let's move to the fact where the permeability depends. So the permeability depends on the number of ion channels that are present, and especially the two important facts the permeability of cell is particularly depend on. First one is the number of channels for that ion. So permeability can be increased by increasing the number of ion channels that are present in a membrane. For example, if we are looking at the permeability of a potassium ion, and if the cell increase the potassium ion channel in the membrane, that cell will be more permeable to the net movement of potassium pretty easily, right? And the second important factor is the ease with which the ions can move through the channels. If an ion is small compared to the size of an ion channel, it goes through easily. But if it's a larger one, it will be trapped. And not only about the ease, because there are two types of ion channels. One is passive, which we are looking in this side, don't have any gate. Another one is the active ion channels contains gate that needs to be opened so that the ions can move. All these factors control the movement of ions across the membrane. Now this permeability can be changed rapidly when they are gated or by any means of gating. For example, they can be voltage gated, or they can be ligand gated, or stress response channels. The permeability of a cell for a given ion increases when gated channels for that ions open. This is the mechanism used for the nervous system to produce rapid change in the membrane permeability. Because in neurons, to transfer that signal from one neuron to the next, they need to alter the resting membrane potential, and it generates what is known as action membrane potential. The action potential is generated so fast, and the only way possible is to open all those gated channels, especially the it's especially the voltage gated channels which are present throughout the axon and axon hillock. Now let's look at the process of the resting membrane potential induction, how it starts. The first thing we talked here is about the change in the membrane permeability and how the membrane permeability will be changed. Well, the first important stage for the generation of membrane potential is the event known as potassium diffusion. So let me change the color once again, otherwise it should take a dark color. This is black, yes. So the first event we can say is the potassium diffusion. Potassium diffuses down its concentration gradient from the cell outside. We know that cell contains more potassium inside and less sodium inside. So if you look at here, we are studying neurons, but before we talk about how ions move across the neural cell membranes, let's talk about the simpler cell, one that is permeable to only one ion. Many of the cells of body are like this simple cell, non-excitable cell. When we understand that, we will learn how the neurons and they function, because they have a different speciality of that function. Now let's say this cell is selectively permeable to only potassium. We are talking about this cell. So selectively permeable to only potassium. So only potassium can move out and in, nothing else. Okay. Now gradients cause ions to move. Potassium will diffuse down its concentration gradient from the area where the concentration of potassium is high to the area where the potassium concentration is low. Now normally potassium concentration is high inside the cell and low outside the cell. So that's the reason potassium will move from the cell outside. So it will diffuse out. Okay. So as this concentration gradient acts as a chemical force that pushes potassium out of the cell, if you look at here, this indicates the direction of the chemical force which is non-excitable cell receives in. So normally a simple movement of it. So if this is the cell we are looking at, and if we think that this blue colored ion is the potassium, we can say that this will move, diffuse out of the cell. This is the event. Now after this event there are some chain events associated. Diffusion of this potassium out of the cell creates an electrical potential across the cell membrane. Do you know why? Because as potassiums are positively charged, movement of all these potassium ions outside the cell cause a net positive charge outside the membrane and a net negative charge inside the cell or cytosolic region. So this separation of charge creates an electrical potential across the cell membrane, which is also provided by this picture. You see mostly negative side in very close proximity of the cell cytosol, and very positive charged surrounding the cell outside. Now this electrical potential will start opposing the diffusion of potassium out of the cell, because once this process start to occur, then what will happen sooner? Potassium concentration will be higher outside the cell and should be lower inside the cell, right? So if you think of this idea, that Imagine that we put our cell in a bath of extracellular fluid and watch potassium diffuse out. As potassium diffuses out, an electrical potential begins to develop. Now, since opposite charges attract each other, the developing electrical potential begins to pull potassium back inside the cell. Because potassium gradient is very high now outside and very low inside. So again, based on the same rule, potassium will tend to flow inside the cell, and this process will continue to occur, continue to go, right? This width of the arrow, if you look at here both the arrow, the width of the arrow, it represents the strength of electrical forces that we are looking. The potassium continues to diffuse down its concentration gradient. The developing electrical potential or force start to increase. Okay. Potassium will continue to diffuse out until the electrical potential is equal but opposite to the force from the concentration gradient. Two forces act on potassium together. These forces are called as the electrochemical gradient. So let's look at that process. These two forces that we're talking about that controls the movement of potassium. One is the concentration gradient or chemical force that causes potassium to diffuse out of the cell. And the second one is the electrical potential or electrical force. This electrical potential originates after the net movement of potassium out and the net change of the membrane potential. And that pulls the potassium inside the cell. And since these chemical and electrical forces on potassium are equal and opposite, there will be no net movement of potassium across the cell membrane. So we can say that potassium here is at equilibrium. So no net movement. The amount of potassium moves outside, the same amount is coming back inside. The force allow them to move outside is the concentration gradient, and the force that will bring it back is the electrical potential or electrical force. Now if we look at the equilibrium potential of the potassium, what we will see? That this electrical potential across the cell membrane is called the membrane potential. That is what known as the membrane potential. That keeps the membrane in a specific shape. The membrane potential is measured in millivolts. Okay, it's a simple voltage, so we'll measure it in millivolt. Because the inside of the cell is negative, the membrane potential is also negative value. And this negative value responds for different cells differently. For example, as we've seen, a simple non-excitable cell has a concentration of 150 millimolar potassium inside the cell, and this is outside the cell. Okay, and 5 millimolar potassium only outside the cell. At these concentrations, the chemical and electrical forces on potassiums are equal and opposite when the membrane potential is balanced at 90 millivolt, minus 90 millivolts, because it's ultimately negative value. Okay. At this spatial membrane potential, the potassium is at equilibrium. This is known as the equilibrium, and it's known as the equilibrium potential for potassium. Similarly, we only saw this example for a non-excitable cell and for only potassium. The same thing can happen also for sodium. The same thing we can also look for chloride, any other ions. Okay, but for the non-excitable cell. But a cell that is excitable, that case, the situations will change. And then we'll see what is the main thing. Now we'll see the electrochemical gradient for sodium and how this thing works. Okay, so let's look at it. We'll recall that the neurons are permeable to more than one ion. So instead of only potassium, we also know that it is also sodium is also permeable. So let's see how the membrane potential in such cells differs from the membrane potential in simple cells that are only permeable to potassium. So when neurons are not generating electrical signals, we said that they are at its rest. They are not generating any signal, but still they are maintaining their membrane potential, right? So resting neurons are very permeable to potassium. So if you look at here, resting neurons, the permeability, let's say this is the permeability high and low, they are very permeable to potassium and only slightly permeable to sodium. They are also permeable to chloride. But since it contributes a little to the resting membrane potential, we should not consider chloride in this purpose. We only consider to talk about potassium and sodium. So we have observed that the movement of potassium ions across the membrane, it's a very permeable cell membrane. Now let's examine the process of movement of sodium. So now if you look at this picture, you will see the arrows are also similarly distributed and the width of the arrow provides the degree of this potential in the movement. Okay, the relative strength of the chemical force. So till now we have learned that the potassium diffuse out of a cell, a charge separation develops. The charge separation is positive outside, negative inside. That is due to the potassium. We saw that. Now, since opposite charges attract each other, the chemical potential resulting from the charge separation, and also we know that the potassium seriously flows in back there. Okay. Now here you see that the width of the arrow again representing the degree or the strength, the relative strength of the electrical force that is there. Now the force from the concentration gradient and the electrical potential, they combines to produce a large electrochemical gradient for sodium. So what happens for the sodium? Okay, let's look at it. Normally the potassium diffuses out of a cell, charge separation develops, producing a net negative charge inside the cell, net positive charge outside the cell. Neurons are very permeable to potassium, so they have a net negative charge inside their membrane. Since opposite charges attract each other, here the electrical potential resulting from the charge separation act as a force to pull sodium also, not only potassium but also sodium inside. That is the simple difference that we see in case of neurons. If a cell is only permeable to potassium, then only potassium should flow. But now we know that not only potassium, but here a lot of sodium is also present outside, and we know that sodium concentration is more outside. So sodium will also flow in along with potassium inside the cell. Okay, and here you see the width of the arrow representing the relative strength of the electrochemical gradient. So we see the electrochemical gradient is very high for both sodium and potassium. That will rush the sodium and potassium inside the cell. This electrical gradient drives the sodium. Okay. Now if you think of the resting membrane potential caused by both potassium and sodium movement, then what we'll find out? In neurons at the rest, the membrane potential is called as the resting membrane potential. So if a neuron were permeable only to potassium, its resting membrane potential would be normally minus 90 millivolt, just like any other normal cells. However, the resting potential here also slightly permeable to sodium, and the electrochemical gradient that we saw for sodium, it also very high, that will move the sodium also inside the cell. The resting membrane potential results from the movement of both sodium and potassium ions, and both of them are positively charged ions, right? So the potential here will be little bit positive than minus 90 millivolt, because now not only potassium is moving in but also sodium is moving in, right? So it shifts this resting membrane potential from minus 90 to minus 70 millivolt. Okay, so if you look at here in experiments, if you look, normally in case of cells which is only permeable to potassium, it should be minus 90 millivolt. But if it's a nerve cell, that case, while the electrochemical gradient is pushing potassium, not only it will put potassium, it will also push sodium. That will change the resting membrane potential from minus 90 millivolt to minus 70 millivolt. Okay, that is how this resting membrane potential will be originated. Now the question is maintenance of this resting membrane potential. You see here it's noted that this potassium, it's relatively large net diffusion of potassium out, and then while we are taking it in, not only potassium but sodium is also taking inside of the cell. So we call it a slight modification. Okay, so what we say in case of nerve cell, the potassium leaks out of the neuron and sodium leaks in. Okay, this is how they maintain the resting membrane potential always. When the resting membrane potential is not equal to the potassium equilibrium potential, the forces acting on potassium are no longer equal to the opposite. Because you see at minus 70 millivolts, if we see at minus 70 millivolt, the chemical force pushing potassium out of the cell, so potassium out of the cell is greater, much greater than the electrical force pulling potassium in. Electric potential, and this is the gradient, right? Now force on potassium is small, but the neurons is very permeable to potassium. As a result, still small amount of potassium moves continuously out of the neuron. But at 70 millivolt, the same situation, but the force on sodium is very large, right? The force on sodium is very large, but the neuron is only slightly permeable to sodium. As a result, a small amount of sodium moves continuously into the neuron. Small amount, but it will move continuously inside the neuron. Okay. So at minus 70 millivolts, the resting membrane potential, if you look at the resting membrane potential at minus 70 and the scenario of both potassium and sodium and what they are doing, you will find out that potassium leaks out to the neuron a little bit, and similarly sodium leaks inside the neuron a little bit. The reason behind it is the permeability of potassium and sodium. Okay, because potassium is forcing inside because of the electrical gradient, the large electrical gradient, and sodium is going inside due to the large electrical gradient, and the potassium is moving outside due to the higher permeability. Okay, now if you look at the same thing listed here, so this is how they keep maintaining the membrane potential. Whatever is generated, that is minus 70 millivolt, it's generated, and once a cell reaches this minus 70 millivolt, we know due to this change in the gradients as well as the electric potential, there is always a net slow movement of potassium out of the cell and slow movement of sodium inside of the cell. Now this is how they balance the membrane potential. But while they need to generate any action potential, they utilizes different types of gated channels. For example, voltage gated channels. You see, whenever they change the voltage gated channels and modify it, it opens up for specific ion only, and that will rapidly change the membrane potential, and that's how the action potential actually originates. We will talk about the action potential in much details in the next video, but for now you should know that the last part of this process is the neuron must compensate for the potassium and sodium leak. And how they compensate for the potassium and sodium leak, and that thing is compensated due to the redistribution of sodium and potassium across the membrane. Because if you look, normally just as a boat that begins to leak will eventually sink, so a leaking neuron will eventually fail to its function. So if the ions leak continues, in that case the concentration gradients of sodium and potassium will decrease. As the concentration gradient decreases, the membrane potential moves towards zero. So when there are no longer any chemical or electrical forces to move ions across the membrane, the neuron cannot send or receive any electrical signal. It needs to communicate. So the captain can keep her boat afloat by simply say belting water out fast as it can, and as fast the water enters. So neurons can prevent the potassium and sodium gradients from running down by transporting potassium back into the cell and sodium back out of the cell. So remember these things. So to prevent this leakage of iron, so the cell needs to transport potassium inside the cell and sodium outside the cell. But the question is, we know that this is against the concentration gradient. Normally, the concentration gradient favors the opposite directionality. So how can cell manage to do that? The answer for that is that cell utilizes a specific ion transporter known as sodium-potassium pump. This sodium-potassium is also known as sodium-potassium ATPase pump. This is the pump which utilizes energy from ATP hydrolysis, help to maintain this minus 70 millivolt of nerve membrane potential for neurons, and also that helps in maintaining the net integrity and the concentration of ions across the membrane, so that the whole process of membrane potential continues. The job of this sodium potassium pump is simply to normally, you see, this is a pump that can bind with both sodium as well as potassium ion. It can also interact with ATP. So two molecules of sodium binds to the specific sodium binding site, and then it will also interact with ATP, and then ATP hydrolysis cause the transporter to open up. Actually, I think 3 sodiums are attached, not 2. So it will open up, then the sodiums are transported out, then 2 potassium will interact, and the release of the phosphate group from the pump cause another structural shift that allows the potassium to flow in. So first the ATP attachment caused the structural shift that caused the sodium to move out. Then the second one, once potassium attached, you see these are, yellow is the potassium and sodium is denoted with this red one. So once potassium is attached, they undergo another structural shift. The phosphate group is released, and that will cause the movement of potassium inside. So this sodium potassium ATPase pump balances the concentration of sodium and potassium across the membrane and reshift the concentration, says potassium ion concentration is higher inside the cell and sodium is higher outside the cell. That's how they like it to maintain. So that in a sense how the membrane potential is generated and maintained. Now if we look at the summary, the concentration of sodium and chloride ions are high outside the cell in the extracellular fluid, and the concentration of potassium and organic anions are high inside the cell. The permeability of a cell for ions depending on the number of type of ion channels in the cell membrane. The electrical and chemical forces for a particular ion combines to become a single force, that electrochemical gradient, which cause the movement of ions across the cell membrane. In simple ion excitable cells, non-excitable cells for example, the membrane potential depends only on potassium. Potassium comes to equilibrium when the membrane potential for the cell becomes minus 90 millivolts. The resulting membrane potential in neurons depend on the distribution of sodium as well as potassium across the cell membrane, and the resting membrane potential in the neurons are commonly around minus 70 millivolt. The sodium potassium pump is essential for maintaining the resting membrane potential in the neurons so that it balances as the resting membrane potential keeps minus 70 millivolt range. Thank you for watching this video. If you like this video, please hit the like button, subscribe to my channel to get more videos like that, and share this video with your friends. In the next video, we'll talk about the action potential and how the action potential is generated and how it's transferred across the neuron. Thank you.